The Nobel Assembly has today decided to award the Nobel Prize in Physiology or Medicine to Mario Capecchi for discoveries of principles for introducing specific gene modifications in mice.
His life story is a testimony to the resilience and power of the human spirit.
His gene targeting truly has changed the course of medical research.
You know, if you have confidence in a particular idea, go for it and see whether you can come through.
Delegates, please give a huge hero's welcome to Dr. Mario Capecchi.
Okay. So first of all, let me say that it's a great pleasure to be here with you, and I wish you all the luck in the world to achieve your dreams. My message is going to be very simple. Believe in yourself and listen to your gut. So if that's all you take with you, that's terrific. That's all I want to hand over to you. My presentation will have 3 parts. The first part, I'm going to tell you a little bit about what we do and how we do it in terms of our science. The second part is a little bit more embarrassing. I'm going to tell you a little bit about my background. And scientists are very comfortable talking about science, but not themselves. If I were really comfortable at talking about myself, I would have become a lawyer. But I chose science. And then the third part is simply something I wish you to aspire to. Somebody in this audience hopefully will be going to Stockholm in the next 10 to 20 years. And I wish you luck in achieving that dream. So that'll be the 3 parts, and let's go for it. So, what do I do as a scientist? Everything is based on this molecule, and I think all of you are familiar with it. This is DNA. And it's a marvelous molecule in the sense that it was built to archive information, and particularly the information to make living human beings. This molecule contains all the information that you receive from your 2 parents that makes you. And the secret of this molecule is right here. It's made up of 2 strands that wind together as a double helix. And you saw in the structure, the outside part is the railings of the helix, and then the steps are actually the information bases. It's a set of letters. Instead of having an alphabet of 26 letters, it's an alphabet of only 4 letters, A, C, T, and G. But in the 2 strands, if one strand has a G, then the opposite strand will always have a C. Or alternatively, if one strand has a T, then the other strand will always have an A. So if we give the sequence of one strand, we automatically know what the opposite strand is. Then those 2 strands can separate, form a template for a new strand to be synthesized, and now you have duplicate copies of that whole information base. There's 3 billion base pairs. If you wrote it out on a textbook, it would occupy 1,000 pages, each 1,000 pages long. I'm sorry, 1,000 volumes, each 1,000 pages long. An enormous amount of information, from going from a sperm to an egg to making a human being. Okay. So we've chose to work with mice, and these are our patients. And the first question you should ask yourself is, why mice? And the reason is that we have a common set of genes with it. With respect to genes, mice and humans are 99% the same. Even though we look very different, we have very different sizes. We have the same developmental plan, that is, starting from an egg and a sperm, the process of making then the baby is identical in humans and mice. We have the same physiology, the same plumbing, the same lungs, the same heart. There are over 30 bones in your hand, and the mouse has the exact same set of bones. The size is different, but the actual set of bones is identical. Okay? So whatever we learn in a mouse will be applicable to humans. And we can do a lot of more experimentation in a mouse than you can in a human. This is the technology we developed. We call it gene targeting. And the upper bar represents your favorite gene, and the asterisk represents a change you made in that gene in the test tube. So what we've been able to do is to take that altered gene, and remember that the asterisk can be anything. We can change one letter, an A or a T or a G or a C, or 10 letters, 10,000 letters, and now we're working on methods to change 5 million letters at a time, maybe even 50 million letters at a time. So that allows us to change the genome of a mouse in any way we want. So we put this new gene into the cell. There's a machinery that grabs it and then searches the entire 3 billion base pairs in the genome, finds the same sequence, and then literally exchanges information with it. So that change we made in the test tube is now in the genome of a living mouse, of a living cell. From that cell, we can then generate a mouse. How do we do that? We use these particular cells. These are called pluripotent stem cells. They are capable of making any cell in your body: skin, hair, teeth, muscle, bones. Given the right directions, they will become those particular cells and contribute to making the embryo. It's these cells that we modify and change a gene in. Okay? So once we've done that modification, we can take those cells, introduce them into a pre-implantation embryo. Okay? So that's showed up above, and the needle contains the altered cells that we changed in the test tubes. Then we introduce it into the embryo, and then reimplant the embryo into the uterus of a living mouse. Okay? The cells that are going in, the cells that are going to make the embryo, the cells sitting right at the bottom of that blastocyst. They're the cells that are going to make the embryo proper. And the cells I'm injecting will participate in that process. And once they're in that mass, they get directions to be told, make muscle, make bone, make nerve cells in your brain. Okay? So they will then know exactly what to do. But most importantly, they also tell you how to make the germ cells, sperm or eggs. Okay? Why is that important? Because then that change that we created in the test tube will then be transmitted to the next generation. And then we can breed as many of those mice as we want and study what happens. And that's shown here. So in the top, you have the needle going into the embryo, depositing the changed cells. We take those cells, make a little slit in the back. Now we can get directly to the uterus, implant those embryos into the mom. They will develop into an embryo and then make a mouse. If we've chosen coat colors that distinguish the cell of origin of the ones that we modify in the cell from those that are used to make the embryo, the blastocyst, then we can see those two coat colors generated in the mouse. For example, brown and black. We'll get a brown and black mouse. We know the experiment worked. And then we can breed to get get mice that are all coming from the cells in culture. So then every cell in that new mouse will have that alteration that we created in the test tube. Okay? And this allows us to study any disease that one can think of. Alzheimer's disease, neurological diseases, heart disease, all sorts of inborn errors of diseases that have caused changes in children. any disease we want, we make that specific change, and we'll have a mouse that has that disease, and we can use it as a platform to study the disease, and once we understand it, use it also as a platform to develop new therapies, all in the mouse. Let me give you an example. Okay. Here's a mouse, an embryo, and what we've introduced is two different genes. One is that makes cells blue, and the other one that makes cells fluorescently green. Okay? We can isolate the fluorescently green gene from a jellyfish that produces it. We can take that gene, put it in the middle of any other gene, and wherever that gene functions, then those cells will turn green, fluorescently green. Okay? Or they will turn blue because of that other gene that we just got from a bacteria. Okay? It's called beta-galactosidase. So what's clear is that we've introduced that gene in a gene that's important for making whiskers. And you can see blue whiskers and fluorescently green whiskers. Okay? And the gene that we went into is called HOXC13. So we know that that gene is likely to be involved in making whiskers. The same gene is also involved in making fingernails. And so what we can see are blue fingernails, and if we put GFP, the green fluorescent protein, we can see green fluorescent nails. Then we can ask, what is the function of that gene? Okay? Now, fortunately, you have two copies of every gene. Okay? You have one that you received from your mother and another one that you received from your father. If you make one defective gene, everything's still okay because the other's still doing its thing. But you have two defective genes, then whatever that gene's doing, it's not doing, and that will have a consequence. And the question is what? And here you see it. Plus plus means two good copies. That's a normal mouse. Plus minus means one bad copy because we've introduced green fluorescent protein in the middle of that gene, and that's still okay. But what you can see this gene is doing is responsible for making hair. These mice have no hair, okay? And that's alopecia, means bald head. Okay? They also have defective nails. Okay, you go and look at the mouse nails, and they're all crinkled up rather than being normal. So we know that this gene is important for making nails. Okay? Now, we can go then to the human population and ask, are there people that are bald and have defective nails? And there are. We can take their DNA and look at their DNA, and they have mutations in HOXC13. Okay? So whatever we found out in the mouse is directly applicable to humans. And this we've done for thousands of genes, and the rule always holds. Occasionally, it doesn't hold. Okay? And the question is then, it says, well, the mouse is a bad model. It actually isn't. There's a reason for that not working the same in a mouse and a human, and by finding out what that difference is, then you find even more about that particular gene and how it's functioning. Okay? So it allows us now to build models of any human disease, study those models, and then develop therapies that then will be applicable to humans. Okay. So now let me go to the next part and talk about early influences. We always start with our mother. This is a picture of my mother when she was 19. She was a poet. She got educated in the Sorbonne and taught literature and poetry. And this is just prior to the Second World War. Then she became politically involved and started a pamphleteering. She joined a group called the Bohemians and started making pamphlets against Hitler, against Mussolini, against fascism, and against Nazism. Okay, she knew that was a high risk, and then she moved up to the mountains in the Tyrol which is way up in the northern Italy, right next to Austria. That's my mother in the forefront. And at that point, actually, she was pregnant, and I was about to be born. In the background is the chalet that she lived, continuing her work with respect to trying to fight both fascism and Nazism with a pen. She knew that was risky business and knew that eventually she would be picked up. And indeed, when I was 3.5 years old, the Gestapo came to the chalet and picked her up. And I knew what was going to happen because in addition to being able to speak Italian, I could also speak German and understand what her fate was going to be. She had decided that it would be much safer going to another family than going with her, so she had sold already all her properties and then given all of that to a farming family, and then that money was then be given to that family, and then I was going to be with them, and they would raise me. And everything seemed to be okay, except that after 1 year, the money disappeared. And that family didn't have money to continue that, so they set me out in the streets, and that was when I was about 4.5 years old. So then I had to fend for myself in terms of both getting food and shelter until I was 9 years old. And I was lucky. I made it. Thank you. Now, in fact, her life was much harder than mine. I mean, she was an attractive woman in a Nazi concentration camp for 5 years. She miraculously, in Dachau, she miraculously survived. And then in 1945, American troops came in, liberated her, and then she spent 2 years trying to see where I was. And what she did was to phone different hospitals and ask, have you ever had a person by the name of Mario Capecchi in your hospital? And she did that all throughout Italy, and then finally, after 2 years of search, she found that there was a person in the hospital. She went there, she arrived on my birthday, and then life completely changed. She got money from her brother, who happened to be living in the United States. He sent money for a boat trip. We took a boat trip and arrived in New York City, saw the the Statue of Liberty, and then for the first time, I arrived on Sunday. That Monday, I went to school for the first time. And made it. It was what I was expecting when I came, and everybody in Europe believed this. They expected the roads in the United States to be paved of gold. And what I found was actually something much more valuable, and that is opportunity. That's what your gift is to you, is that you can make anything you want of yourself if you believe in yourself and listen to your gut. So this is simply a picture in Dachau, where she was inscripted for 5 years. And this is the farm family, the farm where I lived. It actually still exists, the same family still owns it, and I was able to go visit, and I walked around and was able to actually remember where the chickens were kept and so on and so forth, where I spent 1 year of my life, from 3.5 to 4.5. And then once coming to the United States, I moved from a completely asocial world, where essentially I was responsible for myself, to a completely really social world, and that is a commune in Pennsylvania, where 65 families all lived together, owned everything in common. So you can imagine the juxtaposition of going from one environment to that new environment. But that environment was really special in the sense that it was made for kids. We had enormous opportunities, not in terms of goods or clothing or whatever you measure, but in terms of education and in terms of aspirations. We all had dreams. This is my uncle. He was a physicist. He's the person that brought my aunt and uncle to the United States. And he was responsible, for example, development of TV, which he was not very proud of, and no TV in the house. This is myself when I arrived at age 9 in the United States. This is my daughter in the same commune many, many years later. And here is because of the commune, I had an opportunity to work in Mexico in terms of a work camp and helping to build water systems in a Mexican village, 12,000 feet up in the mountains. And then I went to college. I went to Antioch, which I was very fortunate because it has both, something you might think about. One quarter you study, another quarter you then work. Next quarter you study, and the next quarter you work. And the study is associated with whatever you're interested in. If you're interested in theater, you have theater jobs. If you're interested in medicine, you have medical jobs. If you're interested in science, you have lab jobs. And those jobs are all over this country. So in California, New York, MIT, I worked at Sloan Kettering in New York, and so on and so forth. So all of those opportunities integrate what you're studying with what you want to do. That is the actual experiments in terms of working in a lab. This is a picture of Antioch in Ohio, small liberal arts college. Then I went to school at Harvard. This is my mentor. His name's Jim Watson. You may have heard of him, Watson and Crick. He developed the structure of DNA that I just talked about. And then finally, I went to Utah. Why Utah? And that is because it is gorgeous. This is a view from my window in the springtime, in the fall, in the winter, and it's simply gorgeous. And what it allows you to do is to look long distances. I can see over 60 miles in any direction. And that allows yourself to be opened up and be open to new ideas. And then finally, this trip to Moscow. These are the 3 people that went there with me. Martin Evans, Oliver Smitty's, all for developing that technology of being able to change genes in mice. Moscow's beautiful. It's in the wintertime, but all the ocean inundates the whole city. So wherever you are, you can see boats and see beautiful views, many boats. This is the hotel we stayed at, a gorgeous hotel. Here's the actual ceremony. The laureates are in the front. The back row is 50 people that were involved in making that decision. And one reason it's so difficult to predict what they do is that they have to reach a consensus. It's not a popular vote. It's simply everyone has to agree that this year this person is deserving of the Nobel Prize. And there are flowers everywhere. Where do the flowers come from? Italy. Here are the laureates. Here's the royal family. There's an etiquette as to what you should be doing while you're receiving the prize, but it's very simple. You simply watch the king and ask, what is he doing? If he stands up, you stand up. If he sits down, you sit down. And here is a laureates for that year. Here's receiving the prize. And then they have a big celebration. I should add that when you win the Nobel Prize, it's a 12-day party. You get up in the morning at around 6 a.m., you go to bed about 3 a.m. Okay? 12 days in a row. It's exhausting, but it's a lot of fun. And there are a lot of dinners. Here's sitting next to the next queen of Sweden. And then the reason it goes on till 3 o'clock in the morning every night is every night there's a dance. And you have to participate and dance all night long. And here's one example. And that's the end. Thank you.
Yes.
And good luck. And I hope you all reach your
All
dream.
right.
Thank you. George.
Thank you, sir. Come on back. I'll take the clicker. And Eric, come back up. Give it up for Eric. Right over here. And Eric, you take it away.
Yeah. First off, a wonderful talk. It was just really fascinating hearing kind of the full story of how your life kind of started in your humble beginnings and how it evolved into winning the Nobel Prize.
Well, thank you.
So, first thing I want to ask is, what are you up to today? Kind of, what kind of projects have you been working on lately? And
Sure. Right now, we're modeling actually neuropsychiatric two projects. One is modeling neuropsychiatric disorders in the mouse. You might think that the behavior of mice and humans have no relationship to the other, but exactly they actually do. And we can actually, we've just made a model of OCD, obsessive compulsive disorder, and these mice behave just like humans do that have that disease. And people have no idea how these neuropsychiatric disorders function and how they occur. And now we have a model where we can actually directly measure what is the mechanism and then develop therapies. The other project we work on is modeling cancers that are specific to children. Because if I get cancer, I've had my chance. But if a child gets cancer, they haven't had their chance. And they're extremely aggressive, and that's why they're specific to children. And right now, about 85% of the children die, essentially, after 5 years after being first diagnosed. And that hasn't changed for the last 30 years, and it's time that it does change. And I think we need to understand it and then develop better drugs.
yeah, and of course, the work that you're most known for is your Nobel Prize winning work. But has there been kind of other things you've worked on that have maybe not been in the limelight, but you thought were very interesting, very important?
No, I think, we're always striving. The beautiful thing about science is that it's always changing, and we actually embrace change. Many people are afraid of change, where in science we embrace it and try to utilize it for going on. We always build on the shoulders of others. It's not that everybody does something brand new, we're adding to it. And you will be adding to the work that we're doing right now. And so I think that's, and it's, it's a lot of fun. I mean, just all of a sudden you see something that nobody else has ever seen before. And it's that time that really is, it's that eureka moment that we all aspire to, and science gives it to you.
And in your talk, you also mentioned a number of key figures in your life, from your mom to other family members, and then even to Watson. And what I wanted to ask you was then, how for all of these students, how do you think you can develop kind of these great mentor relationships and really find people that can help?
I think that's probably, most important. I mean, there are two things that, for example, when you're thinking about what college is good for you. And what's important is the second part, that's good for you. Each of us are different. Some people like very large colleges, others are more comfortable in small colleges. And what you want to do is find what's comfortable for you and the environment that's going to give you what you need to further your own education. So, and the same thing holds with mentors. Watson was a fantastic mentor, but he wasn't personable, but he had enormous insight. He had vision, and so he provided both opportunity and vision to me. I knew how to do experiments, but he provided me a much broader vision of the world and of science. So again, find a mentor that you're comfortable with, that you can interact with, and that gives you good advice.
And I'm sure that kind of in your research, along with pretty much anyone else's, there's been a lot of setbacks, a lot of challenges. how do you personally kind of handle that? And how do you think is the best way for these students who will be facing those challenges to look at kind of?
There's two parts. Persistence is very important. Stubbornness is bad. And the line between persistence and stubbornness is very difficult to measure. Okay? So you have to know, essentially, if you're not persistent and you fail and quit, then things will never work. But if you continue to do the same experiment and you get bad results and continue to do them, again, you don't get anywhere. Okay? So you have to draw that line, and probably that's the hardest line that any scientist faces, is when to give up a project and when to continue it. In my particular case, I submitted a grant saying I want to do gene targeting and outlined how I wanted to do it, and the reaction was, forget it, it's impossible, no money, period. Okay? So I had to essentially have the guts to take money from another grant, put it all into this, and then hopefully 4 years later I would have results, or else then I would be out of a job simply because 4 years later I wouldn't be able to get a grant. So I had to take a risk, but nonetheless I had enough gut feeling that I was on the right path to be able to do that. And the way you choose that is, it's not easy, simply because you have to have some way of measuring that you're making progress, that you're not going backwards or sideways, but are somehow going forward. And so you have to develop sort of steps to say, this tells me I'm making a progression and going in the right direction. And the other thing that's always important, when you're doing experiments, what you really want to do is learn from the failures. It's not simply that something fails, but you want to know why it failed. And then you learn something, and then it doesn't matter whether you failed or not, because you're still learning something, and then next time you can improve it. So you have to have enough controls in your experiment to tell you why something failed. And then once you know that, then you improve it and then make it better the next time. Whenever I do an experiment, I actually never repeat it. It's a complete waste of time. If I repeat it, it should be exact same result. I always change 1 or 2 variables and see whether that improves it or makes it a little worse. If it makes it better, then I'm in the right direction. If it makes it worse, then I also know where not to go.
Yeah, of course.
Good luck. And good luck.
So how do you think your kind of humble beginnings have changed the way you've received success and received achievements? And I mean, how the world has also kind of seen your achievements because of that?
But, the joy of doing science is actually the process itself. Whether you receive accolades or not is actually immaterial. When we receive a prize, it's not a prize to me, it's a prize to the lab. It's what they've been able to succeed together. And my lab is composed of, first of all, mostly students. Second of all, I have 15 people and around 10 different languages. Okay. So I choose people from all backgrounds and from all over the world. I want people to come with different perspectives, different eyes, then to look at a problem in common. Okay. So I think it's an amalgamation. Where new knowledge occurs is a juxtaposition of things that previously weren't together. And so what you want to draw is lots of different viewpoints, and then from that create something that's completely different.
One last question for you. When it comes to genetics, one of the biggest kind of concerns that everyone has is ethics behind it, and I mean whether modifying these genes is something that's the right thing to do. Yeah. Do you have kind of opinion on that?
Sure, certainly. No, I think when you're doing anything with living creatures, first of all you have to have respect for that living creature. I mean, if I could do an experiment in cell culture rather than a mouse, I would choose cell culture every time. But there are many experiments that I can't learn what I want to learn in cell culture, thereby I use a mouse. But the same thing holds through working with people and any organism, any animal, whatever you want. You have to have respect. But I think what's important is that in terms of ethics, you always are facing ethical. And I worried about what people are going to do with gene targeting. I do it in mice, but I don't do it in people, simply because of those ethical issues. On the other hand, this technology now is being utilized in people, and it's finally starting to work. So they can correct genes and then permanently cure a disease because of correcting the defect, that is the gene itself. Okay, so what you have to do is acquire wisdom. Scientists can provide information, they can provide new ways of doing things, but wisdom requires input from people of all races, of all colors, of all vocations, and then collectively we can then acquire the wisdom and hopefully make decisions that are appropriate to the technology. I think in terms of changing genes in people, one has to be very, very careful, simply because we don't want to make something that's bad. Okay? If I change something in a mouse, that mouse suffers, and that's bad, but at least they're contributing to greater health, and that's how I justify doing those experiments. So we have to think about the problem. We can't simply ignore it, but then we have to weigh the pluses and the minuses, and then under collective wisdom, get to the basis of doing something that's worthwhile as opposed to trivial.
Another round of applause for Dr. Capecchi.